which will come up soon. So we now move from chemistry to biology, plant biology, to our second speaker, and I welcome Christian Fankhauser from the University of Lausanne. He's a professor at the Faculty of Biology and Medicine there at the University in Lausanne, and his research interests focus very much on effects of light on plant growth and development. So while the first talk was addressing light on molecules and then effects in humans, we now go into the plant world. And Christian, all yours, we're looking forward to your presentation.
Thank you very much. Thanks for the invitation. I'd like to share with you my fascination for how light shapes the body and the different organs of plants in the next 25 minutes or so. And I start by using this illustration that was actually used by the Daylight Academy to make a little bit of an analogy between solar panels and plants. You probably all know that plants have this fascinating ability to basically transform solar energy into chemical energy through a process known as photosynthesis. And the major site of photosynthesis happens in leaves of plants. And of course, it's kind of easy to see the analogy between these big, nice flat leaves here, green, and these solar panels shown on the right here of this little slide. And the reason why these leaves appear green to our eyes is because the pigments which absorb, which harvest this light energy, are called chlorophylls, and these chlorophylls mostly absorb in the blue and in the red part of the solar spectrum. Now, plants actually have this ability to transform solar energy into chemical energy, but they also use light as a source of information. And collectively, this phenomena is known as photomorphogenesis. In other words, how does light shape the body of a plant? And this is illustrated in this little movie here, where this dark brown seedling was suddenly illuminated with blue light coming from the side. And you can see that this reorients the growth of this little seedling, a phenomena known as phototropism. And if I want to take my analogy again a little bit with a solar panel, you know that some solar panels actually are equipped with a light sensor, which allows them to follow the sun going through the sky and reorients this solar panel. And that's pretty much exactly what all of these plant photoreceptors do. They basically allow the optimization of photosynthesis. That's one of their key functions. They also do a number of other things, which I will briefly touch upon. One of the best known variables in the light environment that plants care about, it's not only plants, but animals do as well, is the change in day length. Except if you live on the equator, this length of the day changes throughout of the year. And this provides precious information for many plants and animals, in particular, to allow them to reproduce at the most favorable time of the year. I think we all know that reproduction is actually a rather costly thing. And so you have to make sure that this happens at the time of the year when the resources are available. There are other more sophisticated aspects behind this. In some plants where reproduction requires the presence of insects so that they can bring the pollen from one plant to another, you also want to make sure that you make your flowers when the insects are here. So it's a very important timing mechanism. Other changes in the light environment plants have to face on a daily basis is whether you basically have a sunny sky or a sky with clouds. And this has a very large impact on the intensity of light. And many of the adjustments that have to happen in the plant when this level of light changes, and these changes are quite dramatic and can happen very quickly, actually happen at the level of photosynthesis itself. However, various photoreceptors also contribute to the adaptation to these changes in the light intensity, which can be, as I said, very, very quick. A third change in this light environment, and that's the one I'm going to discuss for the rest of this presentation mostly, is actually the information about the light environment gives very precious cues about where the plant is within its community. If the plant is in an open environment, not shaded by other competitors, it will basically get the sun spectrum shown in red on this slide here, which is basically what we get from the sun once it's filtered through the atmosphere down here on Earth. In contrast, if a plant is under a canopy of other plants, this spectrum is changed dramatically, which is mostly depleted in the blue and the red because the chlorophyll pigments that I briefly mentioned before absorb these wavelengths. And so there is much more green and yellow, which is left. And interestingly, and I'll get back to this a little bit later, there is a lot of this far red light. We call this far red. This is red light that our eyes cannot really see anymore, but it's not quite infrared yet. But plants, as you will see, are very sensitive to this far red light. And so this is a very different situation from a cloud, because a cloud might just disappear a few minutes later. But actually, if you have a competitor growing above you, this is something which is probably going to stay. So plants actually react to this very differently. And the way they know it's not a cloud and it's a plant competitor is because this really changes the spectrum of the light environment. So how do plants actually figure that out? Turns out that plants are full of photoreceptors. And this is a list of photoreceptor families present in all land plants, essentially. There is one family of UV photoreceptors. There are 3 distinct families of blue light photoreceptors. This response I showed you in one of the early slides, the phototropism is controlled by the so-called phototropins, which is one of these families of blue light photoreceptors. And the photoreceptors I'm going to discuss a little bit more are known as phytochromes, and they sense mostly red and far red light. Now, you already heard a lot from the first talk about how our visual system works. And when I mentioned the word photoreceptor, there is sometimes some confusion as to what people mean. Often animal biologists or your common person in the street thinks that the photoreceptor is actually this wonderful organ, the human eye or the eye of any animal for that matter. When I say photoreceptor, I mean the actual molecule, the protein associated with a pigment in most cases, which is regulated by the light environment. So these photoreceptors are analogous to the rhodopsin that were also briefly discussed by Dirk Traumer before. So plants don't have a visual organ, but they're full of photoreceptors. And it turns out that you find them a little bit everywhere in the plant, in leaves, in stems, in flowers, and surprisingly, you even find them, at least some of them, in the roots. And if you want to know why, we can discuss this later. And what do these photoreceptors do? This is a little summary. They basically control major developmental transitions throughout the life cycle of a plant. For example, when the plant has a progeny, it's going to be in a seed, and the seed has a little embryo in it, which can be extremely resistant. The seeds can stay alive for hundreds of years in some cases. But once they germinate to become a little seedling, they're actually very fragile. So the timing of this germination is something very important, and the seed takes into consideration a number of criteria, including the light environment. This transition, which I briefly mentioned before, from vegetative growth, when the plant only makes leaf, to reproduction, is another key transition in the life cycle of a plant, because now this plant is going to use a lot of resources to basically reproduce, has to happen at the right time. This is typically what is controlled by day length. But also senescence, the end of the life cycle of a plant, is also controlled by the light environment, by multiple photoreceptors. And this is basically a property given to plants because they can measure light intensity, but also color, and they can measure day length. The responses I'm going to discuss a bit more are what I would call adaptive responses, and mostly this shade avoidance response, which is shown on the right of the slide, where basically depending on whether the plant is in an open environment without shade from other plants or whether it is in a shaded environment, it will really change its morphology. And this is because plants can measure the spectral composition. In other words, this is some kind of a primitive way of color discrimination. And they can also measure actually the direction of incoming light as exemplified with the little movie of the phototropism in the little seedling. So how do plants respond to this shade cue from other plants? There are basically two major strategies, and that depends on the plant species. There are so-called shade-tolerant plants, and they are mostly plants found in the tropics. They can live in the jungle understory. They can live at extremely low levels of light, and there are a number of adaptations, such as very big and flat, thin leaves. But most species, and in particular most species which are agronomically relevant to us, are actually species which are shade intolerant or shade avoiders. Sorry, I need to drink a little bit of water. And these shade intolerant species, illustrated by this little blue guy here, when it's surrounded by these other plants, will try to top over their neighbors. So essentially what we study in my lab is the fight for light, as illustrated in this little cartoon, where all these little grasses here try to grow taller than their neighbor. And this is actually quite a fierce battle out there in nature, which I illustrate with a few pictures here. One I like to show is the strangling fig tree. This is a vine. It germinates on the trunk of a tree, and then it climbs up that tree. And then eventually it shades the support tree so much that sometimes the support tree actually dies. And this is an example that I've seen myself in Queensland, Australia. You don't see a support tree anymore. This is now only the strangling fig tree. And its name is a bit of a misnomer because it didn't strangle the support tree, but it basically shaded it so much that eventually it ran out of resources and it died. Another example is the development of this fir tree here. And you like to have a beautiful fir tree for Christmas, and these fir trees are normally quite symmetrical. But here, if you look at it, this is the main stem, and all the branches are nicely developed on one side, and there is almost no development on the other side. And that's because during the summer, this tree here is green and totally shades this part of the tree. So this part is totally underdeveloped. And this sort of battle for light happens both in agricultural environment, because our land is very precious, so we try to plant relatively densely, but also in natural meadows. Often light is the limiting factor, actually. So what happens? Basically what happens is that you have selective growth of specific parts of the plants. Typically the stem elongates. This is illustrated in a tomato here, the shaded tomato on the right, the sun grown tomato on the left. This actually advantage in the wild to wild species, but for agriculture, this is a bit problematic, and that's illustrated here with these radishes, where if you grow them at too high density, a lot of resources went into growing these parts here, which are called petioles, but then there is almost nothing left in the storage organ, which is actually what you like to buy in the market, so the farmer will have a hard time to sell it. So it does affect agriculture, and it would be interesting to tune this a little bit. So the major strategies to basically get access to more light for the plants are either phototropism, which basically tells the plant in which direction do I have to grow to get the best possible light, controlled by these blue light photoreceptors, the phototropins. And there are this so-called shade avoidance response, which is predominantly a response to the ratio of red to far red light, which is controlled by the phytochrome photoreceptors. So I have my red and blue research team, depending on what their favorite photoreceptor is, and some of them also integrate a little bit. So let me just tell you a little story about phytochrome very briefly. So these are the spectra of phytochrome in its inactive form PR. It's synthesized as PR. And this is actually the light. The photochemistry behind this is also an isomerization of a double bond. And it's a linear tetrapyrrole. In this case, it's the chromophore. And upon light activation, you will have a PFR conformation, which is actually very long-lived. And then you can switch it back with this long wavelength far red light. So related photo switches are now quite desirable in optogenetics to try to get deep tissue penetration and things like this. So you can think of all kinds of application. But for this talk, what you need to remember is that PFR, the far red light absorbing one, is the one which is active. And then you can either go back into the inactive state by thermal relaxation, which is normally pretty slow, or by actually applying this far red light. Now, if we think about this response to shade, what happens is that in sunlight, this ratio of red to far red light is relatively high. It's slightly above 1. So you have a substantial amount of your phytochrome which is in its active PFR conformation. And what this essentially does is that it inhibits elongation of the stem. In contrast, when you are in the shade, you have much more depletion of the red than of the far red. So this ratio drops dramatically. So you lose your active phytochrome. And so now you only have the inactive phytochrome, and it seems that you have some kind of a default state where now the plant grows. Now I have a little movie which summarizes some of the molecular findings that we made that I'm going to show you in the comment. So we're going to look a little bit inside of a plant. So let's first look at a plant which is grown in the sun. And let's go first zoom into the leaf of this plant. And then when we zoom into the leaf of this plant, what we're going to see is what happens in the nucleus of one of these plants. And your phytochrome is in the active conformation. When it's active, it interacts with so-called transcriptional regulators, which are called PIFs. And this interaction actually leads to the destruction of these transcriptional regulators. As a consequence of this, these transcriptional regulators do not induce the expression of their favorite target genes. And one of the consequences of this destruction or inactivation of these PIF transcription factors is that a major growth regulator, which is called auxin in plants, is present at pretty low levels. There is always going to be some auxin because it's an essential growth hormone, but the levels are going to be relatively low. And when the levels are relatively low, growth is relatively modest. In contrast, if you now look at what happens in the shade, your phytochrome is now mostly in its inactive form. And now the PIFs can accumulate and they bind to the regulatory regions of target genes. And I show two target genes here. One of them codes for an enzyme, which is catalyzing the rate limiting step in the biosynthesis of this growth hormone, which is called auxin. So now you produce this hormone, this enzyme, and you then produce the hormone. And another interesting target gene is actually coding for a transporter for this auxin. So you now start to produce much more auxin in the leaf, and through the action of these transporters, which are called pin proteins, this hormone, which is mostly produced in the leaves, is then transported down the stem. And if we now look at what happens within the stem of these little plants, is you now suddenly have much more auxin which flows down. You also have more of these transporters, and this is transported from cell to cell. And what happens if the concentration of auxin gets higher in these stem cells? Essentially, these cells start to elongate, and as a result of this, you will see macroscopically that your little plant gets longer. And that's a summary of some of the things we have done, but many other people contributed in the field, just to get you an idea of how the mechanism goes. But it's actually very quick. You go straight from the photoreceptor to transcriptional regulation. Now, just in the last few seconds, I want to tell you something about what happens actually when only part of the plant is being shaded. And so I go back to my image of the fir tree here, where only this part of the tree was shaded. So we are not brave enough to study this in fir trees because you need a lot of space and a lot of time. So all of our research is done in a very simple little plant, which is called Arabidopsis, which is a weed that you may encounter in your garden. And so we wanted to find out whether we can also see these local shade responses. So what we did is illustrated here on the left. And just with a little LED, we changed the red to far red ratio very specifically just on one leaf here. You can see this little part of the leaf, which is yellow here. This is where we selectively change the red to far red ratio. And if you do this, then we measure the position of the leaf, which is shown on this graph here. And as you can see, the leaf where we irradiated just the tip, you can see the leaf basically went up. So you can imagine my arm is a leaf, you know, this leaf went up. But the other leaves of that plant, they don't move at all. They don't care. So it's really only the leaf which received this cue and actually specifically received the cue on this part of the leaf, which is called the leaf blade, which moved. If you give the same cue to the stem here, actually nothing happens. And we believe that this is probably a good strategy because there is a lot of self-shading happening at the level of the stem, but at this level, not so much. Now, these leaves are actually even smarter than that because you can you can twist them into changing their position, not only going up, but actually also moving sideways, depending on where you change, where you provide the signal. So if you provide the signal at the tip of the leaf, this is what I showed you before, this leaf specifically moves up. But if you now want to look whether it moves laterally, which is shown on the right-hand side of the slide, you see that it doesn't move. But if you change this cue selectively now sidewise, the leaf still moves up, which is a good strategy for these plants to try to overtop neighbors, but it now also moves laterally. So it's almost a bit like a bit of a radar that you have all around the rim of this leaf, and depending on where it gets the cues, it totally reorients its organ. So my little summary is that plants are full of photoreceptors, and one important role of these photoreceptors is to optimize photosynthesis by modulating organ growth and the position of these organs. So that's pretty much it.
Yeah, perfect.
I'll answer questions if anybody has it.
Christian, thank you. That was fascinating to see what plants do with their photoreceptors on there. I think you can end sharing this slide so that
yeah. Let me do that.
discussion people with the larger images on there.
Where do we do that?
And
no, did I stop that? No, stop share. Here we go.
Okay, perfect, very good.
All right.
I would like to start with the question you already kind of mentioned. There are photoreceptors at the root of a plant, but it's dark at the root of the plant. What does the photoreceptor for what it's good there?
Well, there are different ideas behind this. There is a long discussed idea that there is light piping actually. So basically the plant tissue would almost be like an optic fiber. Mm-hmm. It's a bit controversial. It's difficult to measure exactly how much light would go down there. But I mean, as we've seen with the human eye, which is incredibly sensitive, it's actually very much true for plants as well. So it's not impossible. But I think what is more likely is soil disturbances and things like this. You know, a root likes to be in the dark, but there are quite often disturbances of the soil. And then the root wants to hide again. And so it uses both gravity as a cue, but also light as a cue. So I showed you in the shoot, it actually grows towards the light. But if you do the same experiment and look at what the root will do, the root will grow away from the light. And one of the photoreceptors which is present in these root tips is actually the phototropin.
Okay, right, yeah. Then there was a question here from the audience. Can we learn from the principles you have shown us? And they're intriguing how the leaf is moving. Can we learn from these principles how to control, for example, the efficiency of let's say solar panels? So of technical apparatus that also, I think they go for the same goal. They would like to have the maximum exposure on there.
I guess the difficulty is, you know, how much more complication do you have to put to a solar panel so that it tracks the sun? And basically, is it beneficial in the end to add all these levels of complexity so that you can make something where you really have a gain? Of course, you can probably harvest more light, but whether it's worth it, you know, many more pieces could break apart. These things have to work for many years so that it actually is economically viable, and I'm not in a very good position to answer the question. In theory, certainly, and that's probably why many plants will do this. But whether you can practically turn this into your solar panels which follow the sun and something like this, which makes economical sense, I don't know.
Yeah, what I learned from your talk is that measuring the spectral distribution, so in other words, the color of the light on there, is a very important information, and it decodes information. So it could be in principle used to get certain information for shading, for whatever directions, day or month on there. There was another question because somebody worried about what happens with these fig tree after it has overgrown and shadowed the host tree. So did it collapse?
No, they actually live for quite long. I mean, this cathedral fig tree, which I showed in this picture, is in one of the national parks in Queensland. People think it's about 500 years old, but it's a bit of an estimate. So I don't know for how long it has lived without the support tree, but they can actually survive for quite long like that. Okay,
right. So they support themselves now. Some plants, and we notice they don't like so much sun and so much light. So are the mechanism you described for when they hunt for light, the plants, are they then reversed? And you also briefly mentioned mechanisms that plants can do to protect themselves for too much light on there. Maybe you can briefly refer to this.
Yeah, so one of the key mechanisms to protect from excess light, which is a big problem, because actually sunlight, if sunny day, around 40% of it, you already maxed out the capacity of photosynthesis. Mm-hmm. So all the rest of that light, basically the plant has to dissipate, and a lot of it is being dissipated through heat, essentially. Mm-hmm. And that is an issue. And some of these events are actually embedded, I would say, in the greater definition of how photosynthesis works, mm-hmm. With all kinds of sophisticated mechanisms. Another feature is that plants need the light. And so it's like us when we want to go sunbathe, we need to put some sunscreen because we like to go into the light. And so the plants need to basically make their own sunscreen. And so this UV photoreceptor is key there, mm-hmm. because it controls the synthesis of pigments which absorb in the UV. So that basically plants are now capable of not, having the sunlight that they absolutely need, but not suffering the consequences of the UV wavelengths. Finally, regarding a shade-loving plant versus a sun-loving plant, actually at the molecular level, we know very little about it. We know that the photoreceptors themselves are shared. They exist in pretty much all land plants, even the most simple land plants, which evolved on the surface of this planet around 450 million years ago. They have the same suite of photoreceptors. But probably the wiring, and that's often how it works in biology. The components are the same, but the way you wire the different components is different. And so I think what will happen, for example, is that you control to have nice expansion of your leaf, and so that your solar panel becomes very large, but it's very thin. In a plant which is in the sun, you actually have multiple layers of cells in which photosynthesis happens. So you have a gradient of shading within the leaf itself. So maybe the top layers are not going to be optimal because they have too much, but the lower layers are going to do fine. But in a plant which is typically living under a canopy, you will not have multiple layers because the top layer already has a hard time to basically get enough light. There is also a very fascinating plant for which they have described that it basically makes a pigment which looks, I don't know if you see these blue butterflies, which are almost a little bit, when they close their wings, they look very black, and when they open them, it's this beautiful blue. And so this tropical plant has this as well, and this somehow shifts the spectrum of the light a little bit over and optimizes the photosynthesis. So that's one fascinating mechanism. But in general, we don't know very much about those. Mm-hmm. Agricultural plants, it's really, the one I know is coffee, which is not the sun-loving one, mm-hmm. Which is typically planted together with other plants. Most agricultural plants actually like to avoid it, and so.
Fascinating topic, but we have to move on. Christian, thank you very much for sharing this and give us an insight. It's really fascinating what nature has in mechanisms on there. We now change topics.